Okay, so our next speaker is Dahl Jeppesen. She's a PhD student from ARCEDI, Center for Fetal Diagnostic and Department of Clinical Medicine at Aarhus University in Denmark. So it's with great pleasure that I have you once again, Line. Please.
Thank you very much, Joel, and it's all my pleasure. I'm really happy to be part of this really interesting program. Yes, so I'm an industrial PhD student employed by ARCEDI, and I have a grant from Innovation Fund Denmark. I'm also affiliated with the Center for Fetal Diagnostics, where Professor Ida Vogel is my main supervisor. So this is what I will be talking about today. So I will start by giving you a little bit of background about fetal cells in the maternal blood. Then I'll move on to talk about our recent study on cell-based NIPT for cystic fibrosis. Then I will give you an update of our clinical study data where we use cell-based NIPT for detection of copy number variants. I'll briefly touch upon our experiences from the first year of EVITA TEST complete product launch. And then finally, I will talk about the future perspectives and how we see this technology applied in different areas. So ARCEDI is a Danish biotech company focusing on advanced rare cell diagnostics, and the company was founded back in 2005, and since then the scientists at ARCEDI has worked together in close collaboration with scientists at Aarhus University Hospital to develop and optimize a method to isolate circulating trophoblasts from maternal blood. And actually, I want to take you back to the very beginning with the initial studies being done where blood samples from pregnant women carrying male fetuses were collected, and the fetal cells in the maternal blood were identified using the Y chromosome. The fetal cells were pooled for gene expression analysis, and these studies showed that the fetal cells in the maternal blood actually expressed a combination of both mesenchymal and epithelial markers, and this made the authors to suggest that the fetal cell type that we are using might be these extravillous trophoblasts. And this figure here I borrowed from Moses' publication from 2016, and it shows the roots of the routes for the extravillous trophoblast invasion in the first trimester. So here we have the anchoring villi, and in the tip of the anchoring villi are the extravillous trophoblasts. And here they undergo this epithelial to mesenchymal transition and gains invasive properties that allow them to migrate into the maternal spiral arteries and also the veins and uterine glands. And this is how the intact fetal cells shed into the maternal circulation and allow us to isolate fetal cells consistently from maternal blood in gestational age 10 to 15, so in the late or in the end of the first trimester and in the beginning of the second trimester. And these trophoblasts are then the source of the fetal DNA that we can use for non-invasive prenatal testing or NIPT. This figure gives an overview of the technology we use for the ARCEDI technology. So in the upper panel here is the process, and the middle panel gives an estimate of the processing time for each step, and finally in the bottom is the kits or instrumentation that are needed for each step. So after blood collection, we do blood processing, and in this process we get rid of the red blood cells. We fix and permeabilize the nucleated cells, and then we use this specific combination of antibodies targeting a combination of both mesenchymal and epithelial cell markers for enrichment using magnetic activated cell sorting. The enriched cell population is then loaded on a fax for fluorescent activated cell sorting. And when we have the fax isolate the cells individually by single cell sorting, and each cell is then subject to a whole genome amplification. And this product is then used for DNA profiling using short tandem repeats And the detection of paternal alleles, which is not present in the maternal DNA, then allow us to identify the fetal cells. After this, the whole genome amplified DNA from the fetal cells can then be used downstream for variable genetic analysis. And just to mention, as you saw on the timeline in the beginning, this has been a method that has been optimized throughout the last decade and more. So on average, we harvest 10 fetal cells per sample. So this is an extremely rare cell type to work with, which has also been the challenges. But here we are. So now I want to talk about our recent study on how we want to develop cell-based NIPT for monogenic disorders and specifically for cystic fibrosis. And as you're probably very aware of, cystic fibrosis is a common severe recessive disorder. And in Denmark, 1 out of 33 is a carrier of the disease. And we have a newborn screening, and it detects the 1 to 2 babies with the disease every month. Some countries provide prenatal or preconception screening, and a prenatal screening program has been proposed in Denmark by screening all pregnant women, and the detection of a carrier would then open up for screening of the partner, and if the partner is a carrier as well, then invasive testing would be offered. So therefore, we want to, in this study, aim to develop cell-based NIPT as a prenatal screening for cystic fibrosis. And the manuscript has just been submitted to prenatal diagnosis. So this figure here shows the study design. So we had 2 groups of pregnant women, one in the validation group where the women were going through invasive testing for cystic fibrosis due to a high risk of both parents or carriers. And then a screening group where we collected blood samples from 22 consecutive pregnancies undergoing invasive testing for an indication other than cystic fibrosis, and most often due to a high risk result in the first trimester risk evaluation for Down syndrome. So we achieved a cell-based NIPT result in 27 of these cases, and the result were compared with the invasive test sample. We used 2 different methods for the cystic fibrosis analysis. One method was the established fragment length analysis, and then we applied an NGS-based method that we developed in collaboration with a company from the Netherlands called NIPT. And I'll get back to this. So just take a sip of water. So in all 27 cases where we got a cell-based NIPT result, it was in concordance with the invasive test result. And this table here, it compares the 2 cystic fibrosis methods we used. And the study showed that the NGS-based method had some potential advantages. One measure is the lower allelic dropout rate, which is an important measure as the failure to amplify one allele might lead to misdiagnosis. The other things were the possibility to add amplicons for other disease-causing variants, as well as the scalability of NGS. So this is the result of the validation group. So in this group, we identified 3 carriers of cystic fibrosis, 4 normal fetuses, and in one case we did not achieve a result from the first blood sample, as we did not harvest any fetal cells. And I have an example here. Let's see. So in this case, the mother carried this common 3 base pair deletion, F508 deletion. And the father carried a point mutation, this R334W, which is a C to T point mutation. So in the upper panel here is the result from the maternal DNA, and the fragment length analysis is divided into 2 parts. So the part on the left side here detects all the variant alleles and the normal common allele for this F508 deletion. And the variant alleles are detected in the blue color. So here's the heterozygote result for the mother. The second part of this test detects all the normal alleles, And I want to just draw your attention to this peak 10 and 12. You see this represented these split peaks, and this is because she is a carrier of this 3 base pair deletion. Here is the result of the trophoblast DNA isolated from the maternal blood sample. Here we see that the fetus inherited the paternal variant, but the maternal variant was not present. So this is an unaffected carrier. This is the result of the screening group. So in this group, we had 2 samples where we did not harvest any fetal cells and did not achieve a cell-based NIPT result. We had 2 cases where we found the mother to be a carrier, and in all cases, the fetus had a normal result for cystic fibrosis. So this first case, the mother carried this common variant, and she was actually not aware of her carrier status. So the family were referred for genetic counseling at the Department of Clinical Genetics at Aarhus University Hospital. This other case was more spectacular as we identified a relatively rare variant, and I want to show you the results here. So we detected this peak number 9, a relatively rare variant in the maternal sample, and this variant was not present in the fetal sample. The pregnant woman was consulted by clinical geneticists, and in parallel, as I mentioned, we did the NGS-based analysis, and this was surprising because we actually got deviating results. So here in the B part, we see the result of the NGS analysis, and what we see here is that the variant that is present in the maternal sequence is actually 1 base pair upstream from the variant of interest, which is marked here by the blue bar. And this variant that is present is categorized as being likely benign. So this completely changed the situation. And the false positive result here in the fragment length analysis is due to the primer binding, the variant in the primer binding site. And this might occur more often if you wanted to use this method for screening purposes as compared to diagnostics. Finally, in the screening group, we had the invasive samples were tested by ARCEDI, and in the CVS, there was 2 cases with an abnormal result. And retrospectively, we wanted to see if we could also detect these in the fetal samples using circulating fetal cells. And here is in the first case, we had detected a 7.7 megabase deletion at 6q. So here's all the autosomes and the sex chromosomes on the right. And here's the deletion, which is the loss of this 7.7 area in the 6q. Here in the other case, it was a 33 megabase deletion at 5q, and this was also found in the circulating trophoblasts. So in conclusion, the trophoblasts isolated from the maternal blood has the potential to provide prenatal screening for cystic fibrosis and simultaneous screening for copy number variants. And this can be done with a single visit setup. We don't need the paternal sample, and it was with little inconvenience for the pregnant women. I want to give you an update of our clinical study where we compare cell-based NIPT with cell-free NIPT. So since 2018, cell-based NIPT has been added to the current NIPT option in the central Denmark region. It's an ongoing study, and we have more than 400 samples collected, and the final processing of the study data is yet to be completed, but we have published cases continuously. And overall, it shows that cell-based NIPT has some advantages over the current NIPT, which only detects the common trisomies, trisomy 13, 18, and 21. So this is some of the cases where cell-based NIPT have detected microdeletions, which was not detected by the cell-free NIPT. This is a case I want to show you. So in this case, there was a pregnant woman, and she had 3 spontaneous miscarriages. After this, she was diagnosed with mosaic monosomy X. And shortly after the diagnosis, she became pregnant again. And she really wanted a non-invasive test option. So she opted for cell-free NIPT at gestational age 10+3. And as a part of this research project running in the central Denmark region, we performed cell-based NIPT. And here the cell-free NIPT result shown here suggested that was inconclusive for chromosome X, and it suggested monosomy X, as it could not distinguish if the loss of chromosome X were fetal or maternal. And this shows the result of 3 individual trophoblasts and a racegate against a normal female reference. And this case really illustrates how cell-based NIPT can be useful in cases where cell-free NIPT cannot distinguish if the chromosomal change derives from the fetus or from the mother. I think I will skip this case here and move on to another clinical study where we are validating the NIPT resolution on clinical samples. So for the high-risk pregnancies, we perform cell-based NIPT in cases where an abnormal CVS result is shown. So this is the result, and as you can see, it's not only the aneuploidies, but also smaller microdeletion and microduplication syndromes that are detected down to around 2 megabases. This brings me on to the experiences from the first year of EVITA TEST Complete. So EVITA TEST Complete is a cell-based NIPT that was launched on the Danish market last year, and the test is offered on private IVF clinics and ultrasound clinics. We have currently 14 clinics who are offering the tests, and we have detected 2 trisomy 21 Klinefelter and a mosaic sample. So finally, I want to talk about the other applications for the ARCEDI technology. So we hope that we can launch a EVITA test for cystic fibrosis. We have also doing a study in collaboration with the Department of Molecular Diagnostics at Aalborg University Hospital, where we apply cell-based NIPT as a confirmatory test of pregnancies following IVF and pre-implantation genetic testing for different monogenic disorders. And this is an ongoing study. So we are also, as another study we have done was the detection of molar pregnancies, and also detection of or categorization of the molar pregnancy if they're complete or partial, which is important for the prognosis of the disease. We have another PhD student who is doing a study where she wants to use circulating fetal cells for early detection of preterm birth by detection of markers and cells that circulate from the fetal membrane. We are doing a study for monitoring cancer by isolation of circulating tumor cells. And this is our collaborators. I think I mentioned some of them, but these are obviously very important for this research going on. And this is the ARCEDI team, which is really the foundation for all of this research and ongoing optimization of the method. So if you have any question, please contact me. And Yuval, I believe you have some questions right away.
Thank you again. I've been following ARCEDI for the last few years, so it's good to see how it has progressed. I understand about you do have some failure rate. So is it about 10% that you don't agree, or is just for that study?
Yeah, so for the failure rates, which is, I believe you mean the blood samples where we do not harvest fetal cells from the first row, it's about 10%. Yeah.
So the question is about being able to offer it for a large population, and also scalability. Is it scalable to deal with a great deal of samples at the same time?
Yeah, that's a really good and relevant question. So actually, my colleagues are really working at the moment to optimize the method for automatization. So we are in different collaborations to try to build a robot for the first part of the blood processing, and also the magnetic activated cell sorting. And then for the downstream processes, we are moving on to robots. So I believe it's scalable. Then it's for the final genetic analysis. I definitely think that NGS-based methods are more prone for large scale use, which it would be for screening purposes.
Yeah, more robust. So in theory, once you have enough cells, you'll be able maybe to sequence more genes or screen more genes. Is that something that you would consider?
Yeah, for the monogenic disorders, I think it's definitely relevant to look into which variants we wanted to screen for. Definitely, and I think using an NGS-based method, it's also more flexible. With the collaboration we have at the moment with Nimagen, we can always ask them to add in more amplicons for different variants in different genes. So I think it's just a discussion what we want to screen for, basically.
Okay, thank you very much.
Thank you too.
I will soon see you visiting our country.
Yes.
Okay.
I'm really looking forward.
Thank you very much.